My name is Maggie, and my project this summer focused on the signaling requirements of maintaining pig-induced pluripotent stem cells in feeder-free conditions. I know that sounds like quite a lot, so let's dig into it. Previously, culturing iPSCs required a layer of MEF feeder cells that supplied unknown signaling molecules. This aspect of cell culture limited the generalizability and applicability of any results due to some aspects of the cell environment being unknown. Our goal is to simplify the culture conditions of these cells after removing the necessity of the MEF feeder layer. We decided to focus specifically on the necessity of BSA, Activin A, and IWR1, as these are supplements currently being used in our cell maintenance media. My work specifically relied on the use of an OCT4 reporter line. OCT4 is a gene that is required in all stem cells and would function as an indicator of our cell's well-being. This reporter includes a coding region for the fluorescent protein neon green, which will cause the cells to fluoresce green as a representation of OCT4 expression. This green fluorescence allows us to use flow cytometry to analyze OCT4 gene expression by categorizing the cells as OCT4 positive or negative, depending on the green fluorescence level. In addition to this, we also use qPCR and a variety of other methods to analyze the overall health and behavior of our stem cells. My work involves the creation of 12 different medias based on the initial contents of the media that we currently use to maintain these cells. I first investigated the removal of BSA, which functions as a small molecule transporter between cells. Here, we noticed a slight difference in our cell morphology, as those without BSA developed into slightly looser colonies. I noted no change in OCT4 expression when the concentration of BSA was altered based on our flow cytometry data. I also found no significant difference in the expression of known pluripotency markers, indicating the cells were exhibiting similar levels of stem cell gene expression. Finally, I assessed the ability of the cells to differentiate into cardiac muscle cells. This involved a 10-day process following a known protocol that resulted in both the cells grown with and without BSA generating spontaneously beating cardiomyocytes. This indicated that both cell groups showed similar levels of differentiation capability, which is an essential aspect of stem cell behavior. Next, I looked at the necessity of Activin A, which functions as a member of the TGF-beta signaling pathway and is believed to be involved in cell self-renewal. I investigated both the removal of Activin A as well as an attempted rescue by increasing the amount of TGF-beta to determine if that could reverse any effects of the loss of Activin A due to them being in the same signaling pathway. I noted the removal of Activin A resulted in a significant decrease in OCT4 expression, and the attempted rescue with increased TGF-beta lowered this expression even further. The findings from my flow cytometry analysis were then confirmed via qPCR, where I noted a significant decrease in OCT4 and NANOG, but a significant increase in SOX2 expression. So this data suggests that the cells may have actually been differentiating towards a neural lineage. My final experiment focused on IWR1, which prevents beta-catenin from reaching the nucleus, thus blocking the Wnt signaling pathway and supposedly preventing differentiation. We also examined the effect of adding CHR, which is actually a Wnt activator and was expected to function in opposite of IWR1. Based on cell appearances, it seems as though the loss of IWR1 was negatively impacting the cells. When IWR1 was initially removed, we noted no significant difference in OCT4 expression with our flow cytometry data. However, as the treatment continued, we noticed that the removal of IWR1 was associated with the loss of OCT4 expression. You can actually see dual populations present in the absence of IWR1 and CHR, which suggests that some stem cells may be able to function with a basal level of Wnt signaling, but any additional Wnt activation is detrimental, which you can see in media 12. The qPCR data noted no significant change in pluripotency marker expression dependent on the presence or absence of IWR1. However, I believe this to be the case because qPCR data actually represents an average measurement of a collection of cells. So it's likely that the lack in significance is due to the dual populations we noticed during the flow cytometry analysis, as these would average out to no significant difference. To further clarify this, we performed immunostaining to determine cell-level expression of the pluripotency markers. So here we have Hook staining, which indicates the nuclei of each cell and gives an idea of where these cells are in the image area. In the immunostaining for our pluripotency markers, we can see that only our original media 1 shows consistent expression of all three markers, whereas each of the other media show varying degrees of heterogeneity, which indicates some deterioration in their pluripotent state. Following our 3 streams of experiments, we concluded that BSA may not be necessary for maintaining pig iPS cells in feeder-free conditions. However, both Activin A and IWR1 were irreplaceable. Our future tests will focus on the interaction and interdependence of these 3 supplements in maintaining our cells. I would like to specifically thank the members of the CHU Lab for their help in completing this project, as well as the O'Brien Center for supporting my research this summer.